I'm Naomi Rodman from the Crop and Soil Science Department. I am getting a master's in soils research. And my project is in combination with the Wallace Lab and Levi Lab, and we are working with GIS to map microbes in the soil. So this is a really exciting combination of GIS and biological sciences, and it essentially works, we use GIS to make a sample design, and then with all of those samples and using a bacterial spike, so we have absolute quantification, we can extract all of the samples to essentially have stuff that's ready to go off for sequencing. So we would use either 16S amplicon or rRNA amplicon sequencing, so essentially identifying everything that has a 16S fragment. And if we get more funding, we would love to do a kind of shotgun sequencing to see a wider range of who is in the soil. And with this information, we can relate it back to GIS, right, using those technologies to really see who is in the soil, right? And with that information, we can know different things about what different features, how they might be impacting the microbes that are there, whether it's like our field has tile drainage on it, or like another kind of like built lake, or maybe built roads. We can learn a lot from seeing what microbes are where. So this is the Wallace Lab where DNA extractions take place. We obviously have wet benches where we use our kits, and if we ever have to use a bacterial spike as we did before, we use it in our fume hood to keep everything sterile, to make sure we're only growing what we want to grow and only spiking what we want to spike. And to grow out bacteria, so when you get it, they just kind of give it to you in a small little kind of like powder essentially. So you grow it out using media, which you would make from all of those chemicals over there. Mm-hmm. Sorry, I'm that way. Sorry. Measure stuff out, right? Mix it together. It's kind of like a recipe of sorts, and essentially you just put it back out on there, so you only grow what you want to grow. So the work in the Wallace Lab goes from wet lab to dry lab. So with the samples that we extract and all the bacterial work that we do, once we kind of have our wet lab results, you're able to move into the dry lab and work on the computer. And so this includes the kind of microbial analysis work that we do using CHIME, and the GIS work that we do to relate, really actually apply the biology to the real world.